we don't have any vaccines, but on top of everything, this bacterium can be transmitted via vertical transmission. And so that's when a parent that's infected produces an infected offspring. So here's a kind of visual representation of that, where we have a fish that's infected with Psychrophyllum. It's then going to be producing an egg that's also infected on the surface and in the reproductive fluids. And if you zoom in even closer, you can see that the bacterium exists within the perivitelline space, which is the space between the shell and the yolk of the egg. And it's thought to enter through the micropyle, which is that hole up at the top left of the diagram there. And so then this will obviously hatch into a fish that's infected, and then the cycle starts all over again. And so this cycle of vertical transmission is what the real focus of my research is. And so a guiding hypothesis of all of this is that if the vertical transmission of Flavobacterium psychrophyllum is interrupted, then bacterial transmission to offspring will be prevented, thereby reducing the risk of bacterial cold water disease. And so in order to actually break this vertical transmission, we have three main objectives. The first is to develop a novel egg disinfection technique. And so to understand what I'm going to be doing differently with this, you first have to understand what we currently do. And so the U.S. Fish and Wildlife Service, as well as the OIE, they have recommendations for egg disinfection that follow a similar protocol to this, where we have an infected, potentially egg, where it's then going to be fertilized. The sperm will enter through the micropyle, which is that hole that I referred to earlier. We're then going to have a fertilized egg, where it's important to note that the micropyle closes. And then it's at this point that it's going to undergo iodophore disinfection. And this is primarily a surface disinfection, where on the other side we have the disinfected egg that then produces ideally a healthy fish. But if you remember from my vertical transmission slide, Psychrophyllum can be transmitted in the perivitelline space, which is inside of the egg. And so Psychrophyllum isn't the only pathogen that can be transmitted vertically like this, but it is one of the only ones that we know of that can be transmitted inside of the egg. And so under our current egg disinfection protocol, we would have the sperm come in, fertilize the egg, then have the fertilized egg where the micropyle closes. It undergoes iodophore disinfection, but because the micropyle is already closed, that Flavobacterium psychrophyllum inside is untouched, and only any bacteria that exists on the surface will be killed, and we have an unhealthy fish. And so if you're at all familiar with aquaculture and disease prevention, this may sound kind of like a radical idea what we want to do, but it's been piloted in Japan and has some very promising results. So essentially, we're going to have a contaminated, unfertilized egg in a saline solution, and doing this will allow the micropyle to stay open for an extended period of time prior to fertilization. So it's at this point that we want to undergo iodophore disinfection so that that disinfectant can enter into that perivitelline space and kill any bacterium that's in there. So at this point, we'll have a disinfected, unfertilized egg. Then we'll go ahead and fertilize it, allow the micropyle to close, and then produce a healthy fish. And unfortunately, this isn't a matter of just switching around the steps of when you're going to disinfect it. We're also going to be doing this under a number of concentrations and combinations of the actual steps, along with multiple controls to effectively do this, because iodophore at high concentrations can be toxic to the eggs. So we need to determine exactly what amount to use in order to still have a viable egg that's fully disinfected at the end of this. So that's the first step. The second objective in breaking vertical transmission is that we need to create a pond-side diagnostic test for Flavobacterium psychrophyllum. And this is so that it can influence our decisions in real time. And psychrophyllum isn't a new pathogen, and we currently have diagnostic tests for it. However, none of them are quite fast enough. There are issues that we have with all of them that we need to correct for this. And so to do this, we need to ask, what are the ideal aspects of a pond-side or a field-based diagnostic test? And so the first is that it needs to be sensitive and specific. It also needs to yield relatively fast results. And lastly, it can't require expensive or complex equipment, because this is going to be used out in the field at the hatcheries, at the aquaculture facilities, where people don't have access to a lab with all of this equipment that we do. And so if you think about our current options, probably PCR and culturing. PCR unfortunately requires a lot of expensive equipment and a lot of training to effectively do. Culturing is relatively simple, but it can take up to 2 weeks to yield results. And so what we suggest is to use loop-mediated isothermal amplification, or a LAMP assay, to accomplish this. And the reason why is because it really only requires a water bath. It can yield results in 30 minutes to 2 hours, which is much faster than these other diagnostic methods that we have. It's also extremely specific because it's going to be using 6 different primers in order to amplify whatever our target region of DNA is. And then to kind of tie this in with current events, LAMP was actually used for some early COVID detecting as well, which is really cool to be using the same thing there. So to actually produce this diagnostic test, we're going to first have to design primers that are specific to the housekeeping genes in Flavobacterium psychrophyllum, and we can do this using software. So we haven't talked about it a ton yet, and we will, but there are a lot of different variants within the single species of Flavobacterium psychrophyllum. They all have DNA, and so what we're going to be doing is using this common region of housekeeping genes. So it's genes that are required for the cell to operate in order to produce our primers. And so then once we have our LAMP assay in hand, we're going to be assessing how sensitive and how specific it is. And the way that we're going to do this is we're going to have reproductive fluids and eggs, and we're going to serially dilute pure cultures of Flavobacterium psychrophyllum isolates into all of these, and then we're going to be testing it with our newly devised LAMP assay, as well as comparing it with PCR and culture in order to verify those results. And then once this is done, we're going to be taking this LAMP assay out into the field in collaboration with the Michigan Department of Natural Resources during their annual fish health assessments. And we're going to be taking the LAMP assay into the circumstances that it would be used in the real world at the end of all of this. And so we're going to be testing at multiple state hatcheries as well as at multiple private aquaculture facilities in the state, alongside PCR and culture, again, because these are kind of the gold standard of the industry right now. And so we want to verify our results with those to determine how effective it is. So that's the second objective. Our third objective in reducing vertical transmission is to identify the Flavobacterium psychrophyllum variants that cause the most losses in the United States. And there's been a lot of work already done into this, but we want to continue it. So obviously, if we want to prevent transmission, we have to know exactly what it is that we're trying to prevent from being transmitted. And so I've used this a couple of times, my representation of like the variation within this single species. This is a much better representation. I wish that there were only 5, but there are not. As a matter of fact, in the US alone, there's over 100 different genetic variants. And so that's what this is showing. Each number is a genetic variant. Each circle is called a clonal complex, and it's essentially groups of extremely, extremely highly related variants of psychrophyllum, even more so than you would expect from them just being the same species. So why is this important? Well, this anergetic variation in psychrophyllum in all of these different variants can result in variations in the host immune response. And so this means that a single species of fish may or may not be susceptible to multiple different strains of psychrophyllum. And so one way of kind of visualizing this is that it's similar to kind of how we have to get flu shots every single year, because the proteins on the surface of that particular virus, we don't necessarily know which ones are going to be present every single year. And it's similar with psychrophyllum in that the proteins on the surface can vary. And so this means that there's the potential for reinfection if it's not, if we don't have tools to cover all of our bases with this. So to actually identify these, we're going to be using multiplex PCR in order to serotype over 490 US Flavobacterium psychrophyllum isolates. And these are currently in cryostorage. They've been collected over the past 40 years, which is absolutely insane to be working with stuff that old. But so the main goal here is to try to link the genetic side of things with the serotypes so that we know what kind of overlap there is. And so looking forward, we don't want to just publish these findings. We also want them to do good immediately. And so we're going to be, it's super exciting. We're going to be holding a series of webinars and hands-on trainings, COVID permitting, where we'll actually be teaching fish biologists and aquaculture personnel on how to use this LAMP assay, as well as what our new novel egg disinfection protocol is. And then all of this research can also hopefully provide the groundwork for further research into either a serological assay or potentially a vaccine, which would be absolutely huge moving forward. So in summary, as I embark on this, the main goal is to try to prevent losses associated with bacterial cold water disease. And in doing so, we can improve food security, and we can also help with conservation and stock enhancement efforts. Because remember, this disease can cause losses of 70 to 90% of the fish that you're trying to raise. And so the first objective in doing so is developing that novel egg disinfection technique, where we're going to be disinfecting the egg prior to fertilization. Second objective is to create our pond-side diagnostic test. So that's that LAMP assay that's using the housekeeping genes in order to target that specific region in all of these different variants of Psychrophyllum that exist. And our third is to further the great work that's already been done in identifying the Psychrophyllum variants that cause the most losses in the US, and specifically linking the genetic side of things with the serotypes. And then lastly, we're going to be disseminating this work through workshops and webinars so that we can get this LAMP assay as well as the novel egg disinfection technique into the hands of people who can use them immediately. And so all of this hopefully will meet our goal of providing aquaculturists and fisheries personnel with the tools to break the vertical transmission of Flebobacterium psychrophyllum. And so at this time, I want to go ahead and acknowledge the USDA NIFA for being the funding agency for all of this. Thank you to the Symposium coordinators for putting all of this together. I know it was a ton of work. Also, thank you to the Aquatic Animal Health Lab members, past and present, as well as my advisor, Dr. Tom Locke. I was told that we would typically have a group photo here, but COVID isn't the most conducive to all of that. So here's all of us individually, but I'd be happy to take any questions at this time.
Great, thanks Sean for such great presentations. Any question for Sean? Okay, I see one question from Erin. Yeah, Erin, please go ahead.
Thank you, and thanks, Sean. You're probably not surprised about this. I'm interested in these workshops that you're talking about. And I'm wondering if you could talk a little bit more about the people that you'll be working with and what your ideas are for sort of translating the work that you're doing so that you get to that point, like you said, of breaking that transmission cycle.
Yeah, absolutely. So, I mean, of course, this is all very dependent on COVID, but, you know, I'm sure that we can make it work regardless. So a lot of these hands-on trainings are essentially going to be whole day long things. We want it to be an enjoyable event for people who can use it. So it's going to be hatcheries personnel as well as aquaculturists that can put all of this to use right away. Yeah, I mean, I for sure need to familiarize myself, and you know, that's one of the furthest off aspects of all of this, but it's definitely something to keep in mind throughout all of the work that we're going to be putting into this, because I mean, without that aspect of it, there's, you know, not a whole lot of good that can come from just doing this without getting it into the hands of people who can use it.
Any other questions? Right. I don't really see any, even from the chat box. With this, I would like to thank Sean for giving us these presentations, and glad to learn from you. Since actually now we are a little bit ahead of time, why don't we go ahead and take about a 3 to 4 minutes break. Just grab your coffee and take a bathroom break, and we will come back in about 4 minutes. Okay.